So let me just share a brief about VentureStudio. Since 2011, VentureStudio has been supporting a lot of startups. We are sector agnostic and we have supported more than 150 startups. We are a technology business incubator located right in the heart of Ahmedabad city in the pristine campus of Ahmedabad University. And we are approved by the NSTDB as a technology business incubator. And we're also a nodal institute for the government of Gujarat's startup schemes. We have supported more than 150 startups, out of which about 40 of them are in the biotech domain. And we support multiple subdomains in the biotech sector also, including healthcare, agri-tech, diagnostics, therapeutics. And our primary objective is to provide a 360-degree support mechanism to our startups. Now, our support is primarily divided into two categories. We provide support in terms of facilities and also in terms of services. So we have mainly three facilities here at VentureStudio. That is the co-working space, and we have the DST Nidhi supported Prayashala, which is a prototype development lab, and then you have the Birex supported Bionest, which is a bioincubator lab equipped to support startups in the biotech domain. So the co-working space is primarily for those startups who need just a desk space and who can work from their desktops or laptops. But for those who want to do prototype development, we have the Prayashala, which has a lot of advanced equipment for designing, for rapid prototyping, and a lot of machining tools, including 3D printers, electronics equipments, laser cutters, you have VMCs, and many more equipment. And for those who are interested in using the Bioness lab facilities, we have a lot of instrumentation and facilities at the Bioness incubator, including the wet lab space. We have a BSL2 cell culture lab, we have microbiology lab, we have chromatography lab, and we have a huge walk-in cold room if you want to perform experiments at low temperature. And then we have a lot of mid-end to high-end instrumentation, such as molecular biology equipments and FPLCs. We have HPLCs, gas chromatographs, we have freeze dryers, probe sonicators, and much more. So we are not just open to startups, but we are also open to industry. We are open to academia. So we have a lot of students and faculties coming in to use our equipments. And even if you are not affiliated to any organization and you are an individual looking to do some basic R&D for a particular project, you are also welcome to use the facility. And we do a lot of community programs and events to amplify outreach. And we do a lot of other services such as ecosystem connects, business mentoring, support for team building, and IP, legal support, and also guidance for further fundraising. And this particular session today that we are doing for 3D power printing is a part of our BioSkill program series, in which we have a mandate to help promote emerging technologies. And in the past, we've had a very successful session where we brought in a completely new technology called as mass photometry. In fact, in June 2023, we had conducted a workshop in association with BioBeams and Refine UK, which was the first hands-on workshop on mass photometry as a tool for quick, accurate size analysis. And mass photometry is fast becoming the go-to accessory especially for cryo-EM. So just to clarify, mass photometry and mass spectrometry are completely different. They are not related. And if you want to know more about mass photometry, you can check out Refine uh, company's website, REFEYN, and check out their product. So just like mass photometry, 3D bioprinting is fast emerging as a technology that is offering a lot of potential for upgrading innovation in life sciences, especially in the area of cell culture. You have now a lot of opportunity to uh, improve in the areas of advanced healthcare, tissue fabrication, especially tissue engineering, disease modeling, drug discovery. There are a lot of opportunities opening up, a lot of new applications opening up with the advancement of 3D bioprinting. And today we have Sytolab from Bangalore who are going to be talking in detail about this technology and how it is shaping up the future of advanced healthcare. Sytolab was founded by alumni of SRM Institute of Science and Technology, and since 6 years now, they have been into the area of 3D bioprinting, and they also have their flagship product called Bioform, uh, which is an advanced uh, bioprinting technology. They will talk about it in detail in this session. And with Cytolab's mission is primarily to join hands, so join the gap between engineering and life sciences and contributing to the future of regenerative medicine and personalized healthcare solution. So before I hand over the session to Rohan and Chethan, uh, I'm just putting in the information about VentureStudio. In case anybody has any queries regarding the support that VentureStudio offers, you can drop in a mail to our email address. And if you have anything specific to BioNest, you can also drop in a mail to the BioNest email ID. And you can also drop in any kind of questions you have about VentureStudio into the chat box. We'll be happy to answer them. And you are also most welcome to visit our website. So thank you for joining this session, and I will hand over the session now to Rohan. Rohan, shall I hand over to you or Chetan?
Hello, sir. Am I audible? Hello?
You are
Hello?
audible.
Yes, sir. Can I be allowed to present my screen?
Yes, yes, yes.
Yes. Just a second, sir.
Yes, we can see your screen now.
Yes, sir. Lovely. So, a very warm welcome to everybody present over here. Hi, my name is Chethan, and I am one of the co-founders of Sytolab, and we are a deep tech tissue engineering company. And I'll get to what all of that means. I'm assuming a lot of the people here are unfamiliar with this technology, or are here to basically get a good understanding of what it is. So, to go ahead with it, let's provide a little bit of context to understanding what this technology is and the problems that it is aiming to solve. So here I have in front of us, and please feel free to let me know if I'm not audible at any time during the meeting or if my pace is a little too fast. So here in front of you is a graph that shows the data of organ transplants for the past 2 decades in India. So the blue curve that you see over here is the line that denotes the number of people who are put on organ transplant waiting list. For example, by what that means is that this is the number of people every year who require donor organs to survive or to have a better life. And the green curve on this graph depicts the number of organ transplant operations that are done every single year. Now, as you can see, there's a huge gap between the blue and the green curve, and the area under the blue curve is directly proportional to the number of people who were not lucky enough to receive donor organs. Now, here is the second context or the second problem. So, currently, the way we make drugs, this is how the pharmaceutical industry currently develops drugs. So, they have 5,000 to 10,000 different molecules that they have identified to have potential therapeutic value for a particular disease. And over a period of anywhere from 5 to 10 years, it goes a process of in silico testing, in vivo, and in vitro testings to finally then come up with one drug that has pharmacological relevance and no side effects that can come out into the market. And over the years, over the past 7.5 decades, the number of drugs that we are giving out into the market every single year is slowly diminishing. Even though we, as science and technology, is improving further and getting better every single day, we are running out of new and efficient drugs to treat diseases. Now, the cost of development of these drugs has also increased vastly and drastically over the years. And what this basically means is us as the end consumers or the patients or the people requiring these drugs, it gets more expensive to us day by day. Now, what if I were to tell you that there is a possible solution to both of these very large problems that currently plague the healthcare industry today? So, coming to that particular answer, there is a particular technology called tissue engineering or 3D bioprinting. So to understand what 3D bioprinting is, let's first give you a small definition of it, what it basically is. It's an additive manufacturing process that uses cells and biomaterials to fabricate living artificial structures, things that resemble tissues, basically. So to understand how this is made, how is it even relevant, and what are the potential applications of it, we first need to understand a few more concepts that I would like to briefly touch upon. So we are all familiar with levels of organization. We all studied this in our schools. Biology consists of a unitary organism called a cell, many cells come together to form tissues, many tissues come together to form organs and organ systems and complete organisms, so on and so forth. But in mammalian cells or in mammalian organisms, if you were to isolate a group of cells or even a single cell from a particular organism, you cannot expect that single cell to grow back into a complete new organ, a tissue, or a plant, or a complete organism. We can see this happen in plants. This property is called totipotency, and there is a lack of this particular property in humans or mammals. Therefore, we cannot expect to take a tissue or a single cell from a particular organism and expect it to grow back into an organ, thereby solving all of our problems. So, a few researchers a few decades ago understood that a possible thing called 3D printing or additive manufacturing could hold the solution to this particular problem. Now, to understand how it could particularly enable a solution for this problem, we need to understand the context of the whole manufacturing industry first. So, currently, at this moment, how we manufacture anything currently is basically via three main processes. One is called formative manufacturing, where you take a material, change its state from a solid to a liquid, pour it into a mold, give it a particular shape, therefore giving that object its utility. And the same way, we can take a larger object, cut material out of it to give it a certain shape, to give it a functionality. And a newer technology called additive manufacturing or 3D bioprinting came into existence about 5 decades ago, where you add substances to an existing material to give it a shape, to give it a certain utility. Now we know that in this equation, the shape equals to the utility. And in the same way, shape has a lot to do with biology. The shape of proteins denotes the way it functions, its enzymic activities, its drug molecules bind to the receptors based on their shape. So shape is everything in biology. So a few researchers a couple of decades ago understood that if I were to try take these cells that currently grow in a two-dimensional culture well in a petri dish or a cell well plate, suspend them in a matrix that can be artificially created, and then 3D print it into an organ-like structure, then the cells are inherently tricked into believing that they are in an organism and start assuming the functions of the cells that they would initially had in the particular tissue. So there are many different forms of bioprinting, and we'll get to understand the nuances of how it works in the later slides. But currently, or to get an idea of how we can 3D print things, is basically you take a material, for example, in an extrusion-based bioprinting, you take a material that is in a molten state inside a syringe and a nozzle, and then a robot basically moves it in a certain shape, giving it a certain 2D shape and then stacking it on top of each other so that you can achieve a particular shape. The same way we can use a molten material that cross-links or gelates or turns into a solid when exposed to a certain kind of light to then give it a certain shape to then 3D bioprint it basically. Now, basically, what bioprinting is we take any cells and the existing 2D cell culture flow that exists where we do our research currently, we take these cells, we select a candidate material. These are usually hydrogels, which are long chain polymers that have a lot of space for water molecules or water spaces in between that make little tiny homes for the cells to then attach to and start proliferating in. Now, the cells, when they are suspended in this matrix, start behaving like how they would inside the body. Even in the body, we are made up of cells, but it's not just cells. It's cells suspended in a matrix. It's a plasma like protein liquid in your blood. It's an inorganic phosphate in your bones. It is proteins like collagen and fats in your connective tissues, like your skin and your blood vessels. And the same way it exists in the body, what we are doing using 3D bioprinting is just replicating the same process artificially. We take a 2D cell culture, we take a hydrogel, we mix the both together, and then we put it into a 3D bioprinter, and it basically reconstructs the shape that it would have, what would you want to you desire, and then that thing or that scaffold that is created then can perform the functions of an artificial organ or a tissue, depending on how you have selected the biomaterials, what are the cell types that you are working with, and how complex your experiment is. So, coming to the materials themselves that are a very crucial part of this particular technology, there are a lot of wide range of materials being used for wide range of tissue applications. As you know, that the cell types are different in our body, the matrices are different in the body, and therefore, when we try to bioprint different organs or different tissues, we tend to use different kinds of materials. If we want to prototype soft tissues like skin, muscles, endocrine glands, neuronal tissues, we would go for polymers that are biocompatible, like long chain polysaccharides, long chain proteins, things that basically form hydrogels. For example, jelly like materials basically are very good candidates for creating soft tissues. And the same way, to try and prototype hard tissues like bones and cartilages, we would go for inorganic materials that have a lot of mechanical strength, like hydroxyapatite, metal composites, ceramics, bioglass, etc., etc. Now, for each different application, a different material type that suits the cells would be used. Now, coming to the actual process of how this particular technology works or it happens is any 2D cell culture would then, any cells would be taken, expanded in a 2D cell culture, and then a cell suspension would be created from it with a desired concentration. And you would mix this cell solution with a hydrogel or a bio ink of your choice. So the bio ink can be a formulation of one or many different kinds of biopolymers, along with a lot of pharmaceutical agents if necessary as well. For example, if you were to try and create bone tissue, you can start off with a stem cell culture and then introduce things like differentiation factors into the buying. For example, TGF beta 2, which is known to cause an osteogenic differentiation in bone in stem cells, can be mixed in with the bioink, thereby the scaffold that is created slowly starts differentiating into bone cells and a bone-like tissue. And there are different kinds of materials that are being used and different kinds of nanoparticles, different kinds of drug molecules that are being used to create these scaffolds to achieve various different properties. Now, coming to more complex applications of the same, you can use multiple different bioinks with many different cell types to create tissues that are little more complex in nature. For example, if you were to try and create an artificial pancreas, you would first need the islet cells that actually produce the insulin. Now, it's not just the insulin cell producing cells, the islet cells that are responsible for insulin production. Our pancreas also understands the glucose level in the blood to then signal to the insulin cells or the islet cells to then produce a certain amount of insulin. The same way, using 3D bioprinting, what you can do is you can have one bioink in one extruder and one nozzle with the islet cells, and another bioink in another extruder with the endothelial or the epithelial cells that control the secretion of insulin and monitor the blood glucose level. And you can bioprint this into some similar structure that is exists in the pancreas, where the inner cluster of the cells is the islet cells producing the insulin, and the outside is then surrounded by the cells that are supposed to regulate the insulin production in these cells. And the same way, we can create vasculature in tissues where we have a normal functioning tissue and endothelial vascularization cells that are placed in the form of a blood vessel that then turn into blood vessels themselves. And many of the times, it's not a full operation that we have to do as well. We don't have to essentially mimic the entire tissue by ourselves. These cells, when they are present in the matrix, then they start re-engineering the matrix by themselves. They start producing the extracellular proteins and matrices by themselves. They start remodeling the architecture of the polymers inside. And if you were to lay certain cells out, like the endothelial vascularization cells, then they start proliferating in different directions, creating new blood vessels of their own, etc., and so forth. And this here is a depiction of one such experimental process where there is two different polymers and a cell that is mixed to then create an artificial scaffold. So once an artificial scaffold has been created, currently, where the technology stands is it's a research and development tool that is allowing many people in academia and the industry to try and create artificial tissues to try and solve unmet clinical needs. So once you have created an artificial scaffold by mixing a particular bioink with a cell line, what you can do is then understand how the tissue is performing via a series of in vitro tests. For example, the one of the first things that any researcher would study is the viability of the cells in the particular scaffold of the matrix. Over time, are my cells proliferating? Is there an increase in proliferation? Is there cell death that is happening? And once you have established basic viability of the cells in the particular scaffold or the bioink, you would then try to characterize the functions of those particular cells. For example, if you were to try and create a cartilage tissue, one of the key functions of a chondrocyte cell is to be producing collagen at a regular level to remodel the construct or the cartilage itself. You would then study the production of collagen over time in your scaffold to see, is there an increase, is there a decrease, or is it functioning like a cartilage? In the same way, you would test for secretory molecules if you're trying to create an endothelial organ. If you were to try and prototype muscle tissues, you would then check for functions of contraction and retraction so that you can verify that the cells are then behaving like how they would inside a muscle. Basically, it's a very versatile tool that allows you to try and create artificial tissues or organs with certain functions by using different biomaterials and different cells and different processes. Coming to where and all the technology has current applications and its current standing in where it is firstly, one of the major applications of this technology is to be able to create artificial organs or tissues to try and curb the donor organ crisis right now that we have not only in India but worldwide as well. There are a few statistics on my screen that show you how severe the problem is. And a very important aspect of 3D bioprinting, or a very important application of 3D bioprinting that currently exists, is even if you're not able to make organs that are ready to go into people yet, these organs or these artificial spheroids or these artificial organoids have the capability, or they can be used to test and develop drugs on. So currently, how things work is we then identify a couple of molecules via in silico analysis, then we test them on 2D cell cultures, and then we then migrate on to studying the activity of that drug in a mice or a rabbit model. Now, if I am trying to study the anti-cancerous activity of a human anti-cancer drug in an oncomice model, there is a very obvious limitation to my methods. Now, if I had a tumor or a specific tumor from a specific cell from a specific patient that I have created into an artificial tumor in a 96 well plate outside the patient's body, I can then test these drugs on in these tumors, tiny tumors or tiny organoids, and then get far different analysis and data on how my drug is performing when compared to an animal model. and it has a huge application in personalized medicine as well. There is one group out of the UK that is currently using patient derived cells from patient cancers to create tiny organoids or tiny tumor models that then they are testing different combination of chemo drugs on the patient tumors outside the body. What this is basically allowing them to do is to cut down the 3 to 4 weeks of time where a patient would be put on one combination of chemo drug to see whether he's responding well to that kind of chemo, how much is the side effects that are happening, and then the physician would then advise to then switch out one drug for another drug, or to possibly find the best combination of drug that is giving them the most amount of recovery from the cancer. What this allows the researcher or that group to do is to then to reduce all of the pain and suffering that the patient would have to go through to find out what is the best combination of drugs that works for that particular individual. But it also fastens the process, brings in a huge amount of personalization to the medicine that the patient is receiving. So these are a few things that are one of the major applications of 3D bioprinting. And a huge aspect of 3D bioprinting allows us to understand biology itself. It shows us a lot of applications in academic research and scientific research as well. We understand how cells behave differently. The expression of cells in a 3D cell culture changes very drastically to the expression of a 2D cell culture. We understand biology better by trying to study 3D organ models instead of two-dimensional cell cultures. We have applications in artificial meat production as well, where there are scientists who are using fat cells and muscle cells and using 3D bioprinters to layer them on top of each other to create small pieces of artificial meat that can be then consumed and can be a huge boon to how we treat animals today. And coming to the traction or how this technology is being currently perceived and what is at the stage where it's at, is it is being very widely used and tested in academic research currently. There is a graph in front of you that shows the number of 3D bioprinting publications or literature that has come out over the past 2 decades. And you can see there's a very sharp increase in that number. Therefore, it's widely tissue engineering is very widely believed to be the next big step in healthcare and medicine by a lot of different people from this industry. And here are some of the journals that you can go through to find out very relevant work on 3D bioprinting. And these are the Indian institutions that have currently mostly bet on 3D bioprinting. Almost all of the IITs today have large centers of excellences for 3D bioprinting technology, including places like AIMS, ICMRs, etc., etc. And coming to where we stand or how what we are doing in this particular space is Sytolab is a tissue engineering and 3D bioprinting company. So we're not just manufacturers of this particular equipment that does 3D bioprinting. We are bioengineering researchers and scientists ourselves. We have an in-house bio R&D research and development team that can constantly works on applications, different biomaterials, and different cell lines as well. And we come with a very unique approach to this problem is we want to better the technology by collaboration with different academic institutions as well. We understand that where the technology is currently, it's not at the stage to be able to treat millions of different people or to bring a positive change in healthcare. What we believe is the true potential of this technology can be achieved via creating bettering this technology with different collaborators in academia, in healthcare, and many different places. And therefore, we have built a product that overcomes all of these technical problems firstly. So currently, the existing 3D bioprinting systems use a system called pressure-based or a pneumatic extrusion system. Now, that kind of 3D bioprinting only works best for printing low viscosity fluids and not long chain biopolymers that are mostly used in 3D bioprinting. Now, how our machines work differently from the ones that are in the market is the machine first understands the scaffold that you're trying to create. And if trying to create a 5 by 5 mm cube today, it first understands that it will need 3.5 ml of bioink to recreate that structure. And it will then disperse exactly 3.5 ml bioink to then create that structure. And what this basically does is it gives you a huge amount of reproducibility in your tests where you're trying to prototype an artificial organ or tissue. If you create a 5 by 5 mm cube today that weighs 5.12 grams, tomorrow it will weigh 5.12 point something grams or 5.1221 grams. So it gives you a accuracy range of 0.02 ml of the particular bioink solution. And what this basically allows you to do is every time you're working with pharmaceutical agents in your bioink, or you want to control the concentration of your cells in the bioink, this gives you a great amount of reproducibility and reduces a lot of variables in your studies. And we have a lot of bioinks that we have developed in-house, and we have collaborated with different academic institutions to provide to researchers. Our main intentions are to make this technology accessible to many different people in academia and research, so that collectively the technology can grow at a faster pace. And we want to be enabling this ecosystem by not only providing the equipment itself, but by providing the bioink and the know-hows and the technical consultancy required for any researcher to start his research in 3D bioprinting, or to translate his work from 2D cell culture to 3D. This is the extrusion system that I mentioned to you about. It's specifically built for 3D bioprinting applications, and it gives unmatched accuracy in the fabrication of artificial tissues and scaffolds. And we have built a software that essentially allows researchers to not understand the nuances of 3D bioprinting and still be able to fabricate artificial tissues and organs. There are many different aspects of bioprinting or additive manufacturing that has to be learned or understood or acquainted with to be able to 3D print something. And what we have done is we have built a software that understands all of these problems and understands that the users of our products are usually healthcare researchers who don't come from a technical or a software side background, where they understand additive manufacturing and all. Basically, what the software does is it from scratch, from day one, it allows you to fabricate artificial scaffolds without having to learn a lot of XYZ concepts. And we provide a lot of bioinks as well. We have partnered up with Central Leather Institute in Chennai to provide collagen bioink to many of our different clients. And as you know, as some of you might have known, collagen is one of the native ECM proteins that we have in our body, and it is one of the most widely available proteins in our body. And it is a very how would you call it, a very easy plug and play bioink that can be used in anyone's research. If any researcher is trying to understand the efficacy of a drug molecule for a particular liver disease, what that researcher can do is basically procure a collagen bioink, have the same cell line that he's working with in his lab, just mix the two and create an artificial model or an organ model in a 96 well plate, which would give him far better results on how his drug or his pharmaceutical agent or bioactive molecule is performing on that particular liver disorder. So it allows people to move on to or migrate to tissue engineering or 3D bioprinting, or to reap the benefits of 3D bioprinting in a much easier way, adding to how we are positioned in such a way to bring this research ahead and forward. We partner up with many different academic institutions on specific projects on the applications themselves, and to improve the hardware parts of the problem or the bioing parts of the same. And to any all of our clients, we provide an ongoing technical assistance, where we don't just assist you with how the bioprinter works. We help you plan your experiments out, select candidate biomaterials for your application, and if there is a researcher on the other side of the world who works with us or we know of, who has worked in a similar field, help connect that researcher with you to help better plan your experiments and to get better results, validation, data, and literature to bring the possible research out into the clinical phase as well. Now, this is about basically what we do. And I would love to answer any questions about 3D bioprinting or us or its applications, or any wild questions that you guys have out there as well. I would like to thank everyone here for listening to me very patiently for the past 20 minutes. Thank you so much. And the floor is now open for questions.
Yeah, thanks, Chetan. I think that was a very insightful, and you explained it very nicely. And we went a lot into details about the basics of 3D bioprinting, I think, which was necessary. I just wanted to also add that, if you can just tell a little bit about your, current engagements with some of the academic institutions where you are helping them in research projects
Yes.
which
So
are using your
uh,
technology.
So one particular case is there is Central Drug Research Institute in Lucknow that focuses on pharmaceutical products and pharmaceutical testing. So we have partnered up with a researcher over there whose primary work is in diabetes. So he has understood, or he has come to the inference via his previous research, that implantation of brown adipocyte tissues or tissue patches into a diabetic mice has shown effects of diabetes reversal in mice. So we are helping that researcher fabricate the bioink and the artificial scaffold of brown adipocyte tissues or cells that are then being implanted into diabetic mice to then study how diabetes is progressing and how diabetes can be controlled in a diabetic mice model. So that is one example of how we are working with one researcher to try and come out with clinical applications. And we are also partnered up with a few institutions who have created bioinks of their own, and we are helping facilitate that shift of bioinks from their labs to different labs. We are working with IIT Indore on this particular part of bioprinting called fresh bioprinting, which is freeform reversible suspension of hydrogels. It's basically you use a support bath or a hydrogel to then print another hydrogel in. It allows you to create more intricate structures and work with low viscosity bioinks. Those were a few examples as well. And I believe there's a question in the chat box.
Yes, yes.
Yes.
Trivedi has asked a question, Shrum
Mhm.
Trivedi,
Yes.
regarding the. So I think the question is with regards to whether the tissue that is being printed resembles real tissue or they are a
Yes.
copy of real tissues and whether there are any side effects on the body.
Yes. So essentially, the concept goes like this. To create an artificial tissue that would go into a person, we would first isolate the cells from that particular patient himself. So there would be no chances of an immunogenic response because the cells that we are using to bioprint that tissue would come from that patient himself or herself or herself. And apart from that, any immunogenic response that would come up would come from the matrix or the bioing that is used. And the researchers would then work out a way to control the immunogenic response or limit immunogenic responses in the bioink or things that produce an immunogenic response in the bioink. Or then same way of how we would provide immunosuppressants to the patient that also can be used. I think, yes, if anyone would like to unmute and ask any questions, I think that would be
Now,
welcome.
we have one more question from
Mhm,
Sheetal.
mhm,
So again, the question is about biocompatibility. I
mhm,
think yes, obviously biocompatibility is going to be there because that's one of the major issues that
mhm.
currently organ transplant is having. Because
Yes, sir,
as Chetan
absolutely.
mentioned that the cells that are being used for bioprinting, they are your own cells. So there's no problem of biocompatibility. Uh,
Yes, and the materials that are being used to create these bioinks are usually biological materials and sometimes human proteins or human materials themselves. Decellularized ECM is a very commonly used bioink. That is where you would take a piece of tissue from a cadaver or a patient that no longer needs that particular tissue. You would remove the cells from that particular tissue and you would liquefy what remains of that tissue that is decellularized. And that can be used as a bioink. And something that has come out of the body can be used again to go into the body, or there are many different materials that have been studied and understood. For example, biocompatible plastics like polycaprolactone, polylactic acid, hydroxyapatite. These are inorganic materials and organic long chain polymers that have shown long-term FDA approval, FDA approved biocompatibility in humans as well. And a lot of these are currently being used as inorganic implants in patients as well. We currently implant patients with 3D printed plastic cartilages or bone grafts or things to hold tissues together as well currently. Stents as well are used mostly. The materials like Dacron, sutures that are used to sew tissues up, they're all materials that don't come from a biological source but have proven biocompatibility. And materials like that are currently being used in 3D bioprinting to try and fabricate artificial organs or tissues that can eventually save someone's lives.
okay. We have a question from Lopamudra about she wants to know the exact use of bioink slash bioprinter. Uh,
Yes. So the bioink is the material that is used in a bioprinter. A bioink is usually a mixture of cells and a matrix-like material or a matrix-like biopolymer, which basically gives the whole thing a structural integrity. So the use of this particular technology, as I mentioned various times over the thing, is to try and create artificial organs to then donate to people who are in need of artificial organs or tissues. Or these artificial organs or tissues can be used to develop and test drugs on, understand biology at the cellular level, and to do things like personalized medicine, etc., etc.
Okay. Just to add on, I think what I can summarize the way I saw your presentation, and I can summarize it quickly that think of a bioprinter as your normal inkjet printer.
Mhm.
So you provide the printer information about which ink to use, what colors to use, and where to print what. So here you are basically telling the bioprinter what kind of cells to use, what kind of microenvironment to use, microenvironment in terms of what Chetan mentioned, that like hydroxyapatite or hydrogel primarily
Mhm.
that you're talking about, and how to layer that efficiently. You can't do that manually basically because you can't create 3D cells, a 3D cell scaffold manually by layering cells, on a platform like a microwell plate or a tea flask. Because
Yes,
I mean that kind of efficiency and that kind of accuracy can only be achieved when you use microfluidics combined with electronics, and that's what your bioprinter does. Uh,
yes, absolutely.
then you can add on to that if you feel.
Yes, sir. That was a very nice example or depiction of what 3D bioprinting basically is. It's a very versatile platform that allows you to pick and choose different materials and cells to try and create artificial organs. And now, how you create which organ depends on where you place what cell and what materials that you use and the shape that you are desiring to create using a 3D bioprinter. So since it's a 3D printer, it can fabricate any shape that you want, so you can make a heart, you can make a liver, you can make things that resemble bones, things that resemble corneas, different skin tissues, etc. The sky is the limit for this particular technology, and only thing is we need to get better at understanding one of the core fundamental problems of 3D bioprinting, that is vascularization, self-assembly, long-term stability of these bioinks, integration into the host's ECM itself, etc., etc.
Right. We also have a question of, I think somebody is interested in pursuing 3D bioprinting as a career option. I don't know if
Yes,
there are any
absolutely.
specialized courses. I mean, you can take 3D bioprinting as an add-on course to your existing degree, but I don't think there are
So
currently.
currently at an educational institution level, we have very few courses here in India that are full long-term courses that allow students to understand bioprinting. But we have an NPTEL course that teaches bioprinting to students, and in universities abroad, where most Indian students then migrate to do their masters or PhDs, tissue engineering or bioprinting is currently becoming a huge or a very preferred choice for bioengineers and biosciences graduates outside of India. And what I think we aim to or we plan on to be doing is to make sure students here at the BTech, at the bachelor's and the school and the college level understand that there is a novel technology that exists like this that has certain applications, and for us to be upskilling our students and researchers with this technology, because it's just a new technology. There's no concepts that are very new here that have to be learned. It's just a tool that needs to be learned, understood, and mastered to try and apply yourself to problems in the healthcare sector. So I think we have to do we are trying to do a good job at it by organizing sessions like this that make sure that people understand and know that there is a technology like this, and provide enough resources that if you want to pursue this particular technology, be it at a personal project level or at your lab if you're an academic researcher, or at your organization if you work in corporate R&D. So we essentially are doing the evangelization of 3D bioprinting in different researchers and different spaces, and we are very lucky to be having to do this with the Gujarat ecosystem as well. And we would like to thank you very much, Arpit sir, for allowing us to organize this session with the Gujarat ecosystem.
Yes, I think we can do more such sessions down the line. We have a question, very interesting question. I think I was going to ask the same question about regulatory approvals. Now,
Yes.
you need approvals such as so.
Yes.
I think ISO approvals. I think you need for the physical product assets. But what about CDSU or ICMR approvals?
So currently, in terms of our products, we don't need regulatory approvals of CDCSO and different bodies that govern medical devices, because our machines are currently being used for research and development purposes only. So the outcomes are not put into people, or does not have anything to do with patients at the moment. So regulatory affairs don't come in then. But if a researcher were to use our machine to fabricate an artificial organ or a tissue that can then have potential applications in healthcare, then that particular individual case would have to be then presented for an approval or an FDA clearance, and then it can be proceeded that way. And coming to the general objective, the general overview of regulatory affairs when it comes to 3D bioprinting is, since there are living cells involved in the process, then it is a very new thing to be tackling, and the FDA and the different government regulatory bodies, the EU and the EC, are still, there's something called a regulatory lag that exists in many different places, and every time it happens when there's a new technology and the regulatory organizations are not caught up to how to regulate this, how to put rules and regulations on this. So there's still a good amount of regulatory gap that exists, a regulatory lag that exists. But there are a lot of good signs. There is one company out of India that has got an FDA clearance to conduct human trials for hydrogel biomaterial that has potential 3D bioprinting applications as well. And there are US and the EU as well as of this moment. I would love to share more resources about these individual clinical trials that are happening in different places. You can reach out to me post this session,
Yeah, I
anyone
think
if they're
I have put
interested.
the email addresses of both the founders of Sytolab in the chat box. So anybody is most welcome to get their questions resolved. We have a couple of more questions in the chat box. So there, I think
Mhm.
there's one question again from Lupamudra regarding whether you can use this to cure diabetes. So I think Chetan did mention
Mhm.
about the possibility of harvesting beta cells from the the islets in pancreas, and then trying to
Yes.
an artificial
So
pancreas. Uh,
so we can
this is
try
still
and
there's a lot of work in progress. This is at a very nascent stage right now, but the possibilities exist, obviously.
yes, yes, sir. Absolutely, sir. There was one more question I saw regarding the lifespan of a particular
Yes,
scaffold.
yes.
So when it comes to that particular question, there's a choice that a researcher can make or a clinician can make is, do I want my scaffold or artificial organ to remain in the body for a long period of time, or do I want it to integrate, or do I want it to disintegrate or then metabolize in the body itself? That comes with the problem that you're trying to tackle. If, for example, if you're trying to fix a bone defect or fix a structural defect in a particular person or a patient, you would then need what you are creating to have a long-standing effect or long-standing position that keeps the structural defect in place. So you would use materials that don't degrade over time or have very good biostability in that particular case. But for example, if you're trying to fix something like a soft tissue or to try and do something like a wound dressing or something, then cases like that, the material that you're using should essentially be able to disintegrate and then join into the host cells or to be replaced by the host's metabolism uh, to yeah, basically to replace the host tissue, the bioprinted or the artificial tissue with the host's internal tissue. So you would treat it case by case, and there are materials and methods that allow you to control the degradation of these materials over time. You can control the concentration of the things that you initially put in, how bioactively stable they are, how they are treated chemically different. So there are molecules like polycaprolactone, which is a bioplastic that has a very normal biodegradability where it integrates inside the body or it is metabolized by the body over 7 to 8 years. And there are materials that have a few days of biostability inside the body. Different cases, different materials like that. So depending on a case to case, you can then plan and tweak and experiment on the scaffold itself or the material of the scaffold to try and achieve either long-term stability or short-term degradability as well.
I think it's too early to also to comment on the stability of cells because I mean we are still at the lab stage, and once you probably try it on animal models to begin with, at least you can know better about the stability, especially
Yeah,
long
so
term.
there are a lot of yes, there are a lot of materials that are already under investigation and a lot of literature very actively currently being produced. So there are a lot of animal trials with 3D bioprinted organs going on in India and abroad as of the moment. And what different researchers are currently doing is understanding how different materials work with different kinds of cells and different kinds of organisms. So we're at the place or the position where we are understanding the fundamentals of this technology or the building blocks of this technology so that we can then further implement them into applications or solutions.
Yeah, and I think the important point to know understand here is that the technology itself is evolving, and there are many more applications that you as a participant can think of beyond what Chetan has put it here. This
Yes.
field is open to more and more inputs and innovations coming from the academic and the research and innovation community. So there is a lot of room for evolving this sector. We could just take one quick question. There is a question regarding the interaction of the printed organs or the cells with the nervous system after transplantation.
So, where places, if you were to try and prototype or to create an artificial organ that's supposed to go into a person, and if that tissue or scaffold is supposed to integrate with the nervous system for that tissue or function to perform the function that it's intended to, we have naturally observed neuronal regrowth in humans as well. For example, when we transplant limbs or full tissues or full muscles from different people, we have seen neuronal connections that are grown over time as well. So it has to be studied. It's a process that needs to be optimized and tweaked for, and a lot of nuances to be understood underneath it. There are people who are creating direct neuronal tissues using 3D bioprinting. There are people who are integrating it with electronics. There are people who are making circuits out of neurons in bioprinting as well. So it is certainly the possibility is there, but the specifics have to be understood and figured out by people like you who are going to start research in 3D bioprinting and tissue engineering.
Yes, thank you,
I
Chetan,
hope,
so much.
yes,
Thank you
yes.
for this wonderful session. And uh, I'm requesting to all the participants that if you have any further queries, you please reach out to uh, us, both Sytolab and VentureStudio. Put the email addresses again in the chat box. So your feedback is very important to us because this technology is growing, and uh, this depends on the feedback that it receives from the ecosystem, which is you. And we also have we are in talks regarding organizing even a hands-on workshop if possible in the near future. And so this all depends on the kind of feedback that we receive from from the community. So if you're willing to participate in this hands-on workshop, do reach out to us, and we'll be most happy to conduct it here at VentureStudio in Ahmedabad University. So thank you, Chetan. Thank you, Rohan. If you have anything to add, please go ahead.
I think we were good, sir. Thank you so much, everybody, for very patiently listening to us. And please feel free to check out our website. We have a lot of information, and we keep constantly putting posts on the applications and upcoming things in the technology on our LinkedIn handles. Feel free to follow us over there as well, and to stay in touch. And you feel free to reach out to us anytime you want to explore 3D bioprinting. Via if you're an institution, if you're a researcher, if you're just an enthusiast in the particular space as well, would love to have a chat and understand better. Thank you so much, everybody, for your time. Thank you so much, Arpit sir, again for helping us organize this session. It was very wonderful, and thank you all for being a very good audience.
Thank you all for joining. Thank you. Bye bye. Have a nice day.